I'm Anders, and my biology role model is Jennifer Doudna. Jennifer first gained an interest in science after reading a book called The Double Helix in 6th grade. She was impressed by the scientists' ability to work together to solve such difficult problems. During her years in high school, she faced resistance from her peers who made fun of her for being non-native from Hawaii, and from her high school counselor who actively discouraged her from going into a career in sciences. However, she was not deterred and was admitted to Pomona College, where she considered majoring in French but ultimately decided to major in chemistry. Her determination to continue to pursue her goals is inspiring to me because I have and continue to strive to have that kind of academic determination throughout my academic career. Jennifer graduated from Pomona College at the top of her class in chemistry. She then enrolled in Harvard Medical School for her PhD. After working for 3 years as a postdoctoral fellow, she left Harvard and went to CU Boulder, where she studied the structure of RNA enzymes. Enticed by the ability to use a technology called cryocooling, Jennifer accepted an assistant professorship at Yale University, where she worked for some years. She then shifted her focus back to California after both her and her husband accepted and currently hold positions at UC Berkeley. Jennifer enjoys being outside in nature. Currently, she likes to read, solve puzzles, and tend her garden with her strawberry guava plant. Jennifer is involved in a broad range of biological fields such as biophysics, biochemistry, and structural biology. Her recent work focuses on questions involving gene editing techniques such as what are some limitations of our gene editing techniques and how do we overcome them. A major discovery of Doudna's was CRISPR, which is a gene editing technology used to snip DNA segments. In her studies, she is currently focusing on applications and improvement of gene editing techniques in viral insertions. In this experiment, Jennifer and her team used a viral vector to introduce a class of guided endonucleases called TNPBs. They introduced it into a plant with the goal of overcoming cargo limitations of certain viral vectors. In this figure, Jennifer and her team evaluated the editing efficiency of a TNPB-mediated editing using two types of gRNAs. gRNA-12, which targets the promoter region of a gene, and gRNA-2, which targets its coding region. The team modified wild-type plants and plants with a mutation that caused reduced transgene slicing called RDR6. They modified this with the gRNA of their choosing and subjected them to a heat shock treatment or just grew them in room temperature. To create these transgenic plants, the scientists used floral dipping. Floral dipping introduces new genes to a plant's reproductive cells by dipping them in a solution that encourages the uptake of said gene. Jennifer and her team concluded that there is a preference for increased temperature in both target sites and that RDR6 could be used in increasing editing efficiency. In the figure, panel A demonstrates the bar plots of an interference assay that tested the transcript expression of TMPB vectors for cleavage at 26 and 37 degrees Celsius. The purple bars indicate the presence of a sequence of DNA vital to the function of CRISPR, while the black ones do not. Panel B is a schematic of a single expression transcript used for the gene editing. Panel C shows the average editing efficiencies for protoplast experiments using 3 TMPBs. Every dot represents the average editing efficiency of a gRNA. Panel D shows the effective temperature on the editing efficiency of gRNA2 in both wild-type and RDR6 type plants. Panel E shows the effect of temperature on the editing efficiency of gRNA12 in wild-type and RDR6 plants. In conclusion, Jennifer Doudna has made significant contributions to the field of gene editing and is the perfect example of a scientist we should all strive to be. Thank you so much for watching.